So I'm going to speak in English. Is that okay with everybody? Okay. So one thing I'd like to know is how many people here are not full-time biologists? Okay. So it's perfectly acceptable if I lose you to stand up and ask me to explain things so that I don't, so that you're not left out. So, I think you'll find the introduction understandable, but after that it gets complicated pretty quickly. One other thing is that this is shown for all the students here and for some of the professors that we're very, very involved in the large world of innate immunity. And the University of Massachusetts, along with the University of Verona, is putting on a meeting in May. And we'll hope you'll all come. It's called the TOL meeting, TOL 2011. And it's at Riva del Garda in Italy, that's near Verona. And Ricardo assures me that there's plenty of money here at UFMG to send, is there money here to send students and faculty members to the meeting? What do you feel? This would be an intercambio of the best sort. These names here, who I won't go over, are truly the world leaders in innate immunity. They include our own Victor Ambros, who won the Lasker Award. We have Nobel laureates who've attended in the past. And it's an outstanding meeting in an outstanding place. Okay. So the title of my talk today is DNA mediates inflammation in malaria. First, let me give you an idea of what innate immunity is. Innate immunity is the immunity that we are born with. So that's the type of immunity that you don't get from having a vaccine or having an infection. It's what you start with before you've ever experienced any other infectious illness or infectious stress. So take this, for example, as to why we need innate immunity. E. coli, which is one of the most common bacteria in the world, once it gets in your blood, can double every 30 minutes. So for example, if you didn't have any way of fighting your first infection, you would soon have 2 times 10 to the 43 bacteria in your blood in a period of just 3 days. It's the adaptive immune system, that's the one that we use when we give people vaccines. It actually takes at least 3 days, and more like 14 days, to have a real response. So having 2 times 10 to the 43 bacteria in your blood is incompatible with life unless you're born with some type of innate immune system. It's undeniably an important part of our complete immunology. And let me give you a few examples of people who have genetic lesions in innate immunity and what it does to their lives. Here's the 2 proteins that are involved in the toll signaling pathway, which I'll talk about later. MYD88 and IRAK4. People with these deficiencies have been identified, and they have recurrent pathogenic bacterial infections. These are a real problem during infancy and early adulthood, but as acquired immunity takes over, these go away. UNC93B is a protein that regulates intracellular toll-like receptors that are responsible primarily for viruses, and TLR3 is a receptor for RNA viruses. Both of these deficiencies have been defined and have been associated with recurrent herpes simplex virus encephalitis. The IL-12 receptor is very important for our ability to fight mycobacterial infections, and people who have defects in IL-12 receptors have recurrent mycobacterial disease. It's very resistant to chemotherapy. It can also work the other way. Not having a perfect innate immune system seems to be good for some people. There's a toll adapter molecule known as MAL, also known as TRAP, and if you have a mutation in this molecule, it's been reported that you have 4-fold reduced death due to malaria, that would be cerebral malaria, and 2-fold reduced death due to tuberculosis and invasive pneumococcal disease. So if you get the right type of mutation, you do very well in the innate immune system. This is Christian Nusslein-Volhard, who won the Nobel Prize in Medicine in 1995. Nusslein Volhart is not an immunologist, she's actually a developmental biologist, and she was interested in the development of the fruit fly, Drosophila melanogaster. What you're looking at here is a mutant that she discovered in the early 1990s, or actually the late 1880s, in a Drosophila. This mutant has what's called an abnormality in dorsal ventral patterning, so that at an early stage of its life, instead of the cells in the body lining up the way they're supposed to be, all the cells think that they belong on the dorsal surface of the organism. So in fly lingo, this animal would be known as a fully dorsalized mutant. What Nusslein Volhart discovered, and was one of the reasons she won her Nobel Prize, is that these mutants had a defect in a receptor that she called toll. And this is a receptor that was involved in sending signals to cells that they wanted to go to the dorsal side of the cell or the ventral surface of the cell. Now, she had a student working in her lab at the time. Her name is Catherine Anderson. She's actually now a dean in New York City. And Catherine's an American and was working in Germany and came to the United States after working in the Nusslein Volhart lab and established her lab at Berkeley. And together with David Schneider, they cloned toll. And when they were looking at the amino acid sequence of toll, they made a very important observation. And that was, toll seemed to have homology to an immune receptor, human interleukin-1 receptor. And it was in thinking that through that they said, well, perhaps toll isn't just involved in development. Perhaps toll is also involved in immunology. That thought was picked up by Nick Gay, a professor at Cambridge University in England, who published a paper who suggested that toll was a very important central player in innate immunity. So this became a hot topic in immunity at the time, and a young graduate student, shown here, Bruno Lemaitre, and his professor, not shown here, Jules Hoffman. That's what happens as you get older. Famous professors just fade away. I don't know where Jules went to. Anyway, this is Bruno here, and he obtained some mutants in toll from the Nusslein Volhart lab. And these mutants, using some tricks, can be actually matured into adults that don't have the receptor. So this is a fly, a toll mutant, that's grown up into an adult. And what Bruno's doing here is he has a very, very thin glass pipette, and he's dipped that pipette in Aspergillus fumigatus, which is a fungal organism that causes death in immunocompromised hosts. And you can see he's inoculated that fly in the pneumothorax. And should those flies live the inoculation, a small number of them die just from being poked in the chest. Well, what will happen to them is that they'll ultimately develop an infection. So in the case of the toll mutants that have been inoculated with Aspergillus fumigatus, you can see that the animal has been overwhelmingly infected with fungus, and the fungal hyphae have grown out of the thorax and killed its dead, whereas in wild-type flies, this doesn't happen at all. Toll mutants also have abnormalities in their response to gram-positive infections, but not gram-negative bacterial infections. We now know that there are 10 human TLRs, 13 mouse TLRs, and quite amazingly, there are organisms that have even more TLRs. So this is the purple sea urchin that lives in estuarial waters. These are filled with bacteria and microbes. These animals live about 70 years. They only have an innate immune system. They have no ability to make antibodies. They have no lymphocytes, etc. And yet they live because they have a very, 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 very advanced innate immune system, advanced through the processes of evolution. All toll receptors have, in the extracellular domain, that's the part that sticks out of the cell, something known as leucine-rich repeat motifs. And these leucine-rich repeat motifs are very important for binding things, the types of ligands that activate the toll receptor. And of course, in order to be a toll receptor, you have to have homology to a certain area of the human interleukin-1 receptor. And that area turns out to be in the cytoplasmic face of the toll receptor, and it's known as the toll interleukin-1 resistance domain, or some people will call it the toll interleukin-1 receptor domain. Now, in addition to these transmembrane proteins, there are 4 toll adapter proteins. Those are known as MAL, MID88, TRAM, and TRIF. That's MAL, MID88, TRAM, and TRIF. And most immunologists like to break the adapters into 2 groups. The first group we consider to be the MID88 dependent TLRs. Those are TLRs like TOL2, which signals through MID88, TOL5, TOL7, and 9. And then we think of the MID88 independent TLRs. The only one that's truly independent is TLR3, which signals through the adapter known as TRIF. TLR4 is the odd player. This turns out to be the LPS receptor. And TLR4 uses both a MID88 dependent pathway and a MID88 independent pathway. Now notice that if we had a mutant in which TRIF were knocked out and MID88 were knocked out, that we would have no toll receptor signaling whatsoever. And we'll come back to that a little bit later, because we actually have such mice that don't have MID and they don't have TRIF, and that means they have no functional toll receptors whatsoever. The other way we like to break up tolls is to talk about them as endosomal and surface TLRs. And the slide is gone. I think we had the file became corrupted when we transferred it from the Macintosh to the PC. Colonel?
Yes.
Can you bring up that slide with me again? Which one? Can you bring up your slides that had me and you had my slide? That's it. This is the missing slide. Fortunately, my picture lying on the gutter is not part of my talk. So we have MID88, we have endosomal and surface TLRs. So a surface TLR would be one, of course, that sits on the cell's surface. And the best example of that is toll-like receptor 4, also known as the LPS receptor. And it sits outside the cells looking for bacterial products to tell the cells that there's been bacterial invasion and to activate the cells. Another group of receptors sits in the endoplasmic reticulum. Those would be toll-like receptors 3, 7, 8, and 9, and in the mouse probably also include 11, 12, and 13. But we're not really going to be talking about mice today, so we don't have to worry about those. So, and the best understood of these is TLR9. In the endoplasmic reticulum, in addition to these TLRs, is a protein known as UNC93B. And UNC93B actually regulates the endosomal TLRs. So what we know about the endosomal TLRs is that under normal conditions, they sit in the endoplasmic reticulum. But when DNA or RNA gets incorporated into an endosomal compartment in cells, they translocate from the ER to the endosomal compartment, where they can then bind the ligand inside the endosome and initiate an innate immune response. So, since these endosomal TLRs have an intracellular location, it's not surprising that they are very important for certain viruses. An example of this is TLR9 is responsible for herpes simplex virus. TLR7 is responsible for human immunodeficiency virus, and TLR8 is responsible for responses to influenza. This is an experiment that I borrowed from a paper by Akiko Iwasaki from Yale, and this was published a long time ago. And what Akiko did was she took cells from certain mutant mice, MYD88 knockout mice and toll-like receptor 9 knockout mice. So MYD88 is the only adapter used by toll-like receptor 9, and she stimulated those cells with DNA that was rich in CpG motifs or ultraviolet inactivated herpes simplex virus 2. And when she does that, as you can see in wild type, WT here, you can see that herpes simplex 2 and CpG both activate an interferon alpha response. In toll-like receptor 4 knockouts, we see the same phenotype. But in TLR9 knockouts, or in knockouts that are missing the adapter for TLR9, there's no response to DNA or deactivated virus whatsoever. We like to think of toll-like receptor 9 as the prototypical endosomal TLR, and the reason we think of it as prototypical is it's the one that we have studied the best. There may indeed be differences between TLR9 and the other endosomal TLRs that we don't appreciate. We didn't really have any idea what the function of TLR9 was until several years ago, when Shizuo Akira, shown here, characterized his mouse that was deficient in TLR9 and demonstrated that it recognized DNA that was rich in CpG motifs. Now, CpG motifs are motifs that are very highly prevalent in bacteria. And it was with this observation that we, in my laboratory, began our work. This work that I'm showing you here was done by Eike Lotz. And what Eike Lotz did was demonstrate what I just told you, that TLR9 moves from the endoplasmic reticulum to the endosome to encounter its ligand. Now in this case, it's not a virus, it's an oligonucleotide that's very rich in CpG motifs. This is a mouse dendritic cell, and it's been infected with a virus that encodes for a fluorescent form of toll-like receptor 9. The cells have been incubated in fluorescent CpG DNA, so everything that's red is DNA, everything that's green is TLR9. And if we look at an endosome, blown up here on the right, we can see that 5 minutes after the cells have been incubated with DNA, there's DNA in the endosomal compartment. And now at the same time, we can see toll-like receptor 9 in the endosomal compartment. So this translocation event happens extremely rapidly. We think that similar mechanisms occur for TLRs 3, 7, and 8. Now, a quick switch to malaria. I'm not going to do a big introduction on the epidemiology and demographics of malaria, since there are entire talks about this, but it's fair to say that this is a very important disease for everyone, not just economists and demographers, but for innate immunologists alike. The disease begins with the inoculation of sporozoites by the Anopheles mosquito. These sporozoites travel to the liver and begin asexual replication there. In time, schizons in the liver rupture and release merozoites into the blood system. These merozoites then have the ability to attach to red blood cells and invade and go through cycles of asexual replication. And very rapidly go from being in very small numbers in the blood to very large numbers in the blood. And it's at the time that the schizons rupture and release merozoites into the blood that patients experience symptoms. The clinical presentation of malaria often begins with what's known as a paroxysm, which can be defined as a fever up to 104 degrees Fahrenheit or 40 degrees centigrade, rigor, headache, myalgias, and other systemic symptoms. During this paroxysm, extremely high levels of the cytokine tumor necrosis factor alpha have been measured. And after the paroxysm, it is said, although I've never experienced the paroxysm, it is said that it's followed by a period of extreme fatigue. And I suspect it's during this period that the patients whom we've had the pleasure to see in Porto Velho, in Dr. Hilbrando's clinic, that they get on the bus and they travel to the clinic to find a doctor. So the question in our laboratory that we wanted to approach was, what's the origin of the cytokine storm that causes the fever in malaria? Now, our initial hypothesis, because in those days we thought everything was caused by TLR, our initial hypothesis was that fever was caused by the activation of a toll-like receptor. In that regard, we thought that there was a real possibility that the inflammation in malaria was similar to that caused by molecules that were LPS-like from gram-negative bacteria. So just let me take a short step back and say that we know a lot about how gram-negative bacteria activate the innate immune system. Gram-negative bacteria shed LPS, or endotoxin, and that LPS molecule binds to a protein known as MD2, shown here in its crystal structure. And if we look at this very closely, these green and blue circles here are the fatty acids on the lipid A portion of LPS that have bound to MD2. Now what happens in TLR4 signaling is MD2 is bound to toll-like receptor 4, and as a result of all three of these molecules coming together, we have a dimer interaction. And that is the same as saying that we have an activated receptor. So LPS is shed, it binds to MD2. This combination of LPS-MD2 binds to TLR4. There are changes in structure that result in dimer-dimer formation, and signaling occurs. So our initial hypothesis was that something like this happened in malaria. And indeed, malaria does have a molecule, the glycosyl phosphatolynositol anchor, that has some structural similarities to lipid A. It has these fatty acids. It has this this carbohydrate attached to it. So we thought, well, maybe the GPI anchors are responsible for activating cells in malaria. Indeed, several years ago, Ricardo's lab and my lab looked at GPI anchors from Trypanosome cruzi, and these were toll ligands. They activated TLR2. And years later, Chang Gowda's lab looked at GPI anchors from Plasmodium falciparum and found that these also could activate toll-like receptors. So the concept that TLR2 was the LPS of malaria seemed simple, it seemed great to us. We really bought into it very rapidly. It was so simple that really anyone could understand it. Well, maybe not anyone, but most people could understand it. But like everything else that Sarah Palin seems to understand, it was wrong. Indeed, it doesn't turn out to be true. And we know this pretty much now, although we've assumed it for a while now. And that was shown very formally early this year when Chang Gowda's group in Pennsylvania demonstrated that the concentration of GPI on the surface of merozoites is too low to account for the ability of the parasite to stimulate cytokine production. There simply isn't enough GPI anchor in malaria to cause fever. So GPI is not likely to be an important cause of cytokine activation during malaria. Well, that led us to look for other candidates, and at the time that we became convinced that GPI was not important, a postdoc in my lab, Fani Lau, was very busy working on a crystal known as hemazoin, here shown in the parasitophorus vacuole of a malarial parasite that has parasitized an erythrocyte. Hemazoin is often referred to as malaria pigment, and that's because people who have malaria, from overwhelming malaria, often have very heavily stained tissues. This is a spleen from a child who's died from overwhelming malaria, and you can see it's now purple. Again, Shizuo Akira comes into the picture. Dr. Akira is actually one of the truly great scientists in the innate immunity world, and he publishes this paper just as I would say Fani was preparing to submit her paper to the Proceedings of the National Academy of Sciences. Dr. Akira publishes this paper in JXMed. Toll-like receptor 9 mediates innate immune activation by the malaria pigment hemazoin. And in this paper, Dr. Akira suggested that hemazoin was the first non-nucleotide ligand for toll-like receptor 9. And that left Fani running out of the lab. Again, we have been scooped by Akira, and she has since, I'm sad to say, left the world of science. But the project didn't die right there, because a very talented technician in the laboratory, Peggy Peroche, began to look at hemazoin more carefully. And what she did was she first went and got some knockout mice. Dr. Akira gave us all of his knockout mice. And she found that when she took dendritic cells from these knockout mice and hemozoin that we had purified from cultures of malaria in the laboratory, that hemozoin really did not stimulate TLR9 deficient cells, nor MYD88 deficient cells to produce cytokines. Here we're looking at the stimulation of IL-12P40. In contrast, wild-type cells were activated and TLR2 knockout cells were activated. So in this respect, these experiments confirm Dr. Akira's paper. But here's where we differ from Dr. Akira, and that is Foundy found that when she pre-treated hemozoin with DNase, an enzyme that destroys all the DNA that's present, that the ability to stimulate cytokine, or in this case chemokine production in cells, is dramatically reduced. Here shown in red and blue, these are two types of nucleases that have been used to digest hemozoin. And the rest of the data on this slide is merely controls, and they're giving the expected results. So we concluded that the cytokine-inducing activity of hemozoin was really due to DNA coating the surface of the crystal. And in fact, when we looked to see what the source of that DNA was, it appeared to be the parasite itself. And that's shown in this experiment where we used hemozoin as a template in a PCR reaction. And we have primers that are specific for Plasmodium falciparum or pan-Plasmodium species primers. And using hemozoin as template, you can see that we PCR out a product. If you do the same experiment using human primers, there's no product, and the rest of these are controls. So the source of the DNA was from the Plasmodium itself. And if one takes plasmodial DNA and using a little trick that we use in the laboratory, we mix it with a transfection reagent called TOTAP that puts the DNA into the endosomal compartment, you can see it's very nice, it's stimulating cytokine production, but if you look at the TLR9 knockout cells, there's no cytokine production whatsoever. So that is where we think the TLR9 dependent component of hemozoin stimulation comes from. So what does hemozoin do? Well, one very important feature we think is that hemozoin functions to traffic DNA into an intracellular compartment to which TLR9 can be recruited. This is Parisa Kalantari, a postdoc in my laboratory, who has been studying this idea. And Parisa's taken synthetic hemozoin, and she's coated it with fluorescent oligonucleotides and fed it to cells. So these are actually macrophages that have been fed DNA-coated hemozoin. You can see the hemozoin inside the cells, and now you can see the DNA inside the cells. And if you do the same experiment, but stain the lysosomal compartment with something called lysotracker, which is blue here, you can see that the lysotracker, the hemozoin, and the DNA all overlay with one another, showing that the hemozoin has taken the DNA and put it into a lysosomal compartment where signaling can potentially occur. Now, if you do the same experiment and the hemozoin is actually bound to the DNA, so you add the hemozoin separately, it gets into cells, it gets into the lysosomal compartment, as you can see. You add the DNA separately. When we look after 30 minutes, the DNA hasn't even gotten into the cells. So everything else gets into the cells but the DNA. So the DNA has to be bound to the hemozoin in order to take a free ride into the lysosomal compartment. Now that was all said and good. We published a paper on this. But as we were doing so, we were quite aware that CpG-containing DNA is not common in the malarial genome. In fact, the P. falciparum genome is the most highly AT-rich genome that's ever been resolved. And in particular, it has a recurrent motif, A followed by 5 or 6 T's, and A and a C. And thanks to the work of Daniela Bartholomew, we now know that that's present in the P. falciparum genome over 6,000 times. Indeed, if one compares various species of microbes and even humans, we find that this motif is very common in biology. There's Plasmodium falciparum, there's Homo sapien 268,828 times. So this is a very, very common motif. And we thought that there was a possibility that it might be immunologically active. So several people in the lab, especially Shruti Sharma and Peggy Perosh, designed oligonucleotides that had this motif in it and used a transfection reagent to get the motif into the cytosol of cells. When they did so, they found that the motif, that oligonucleotides containing the motif activated cytokine production such as TNF, IL-1, IL-6, and interferon beta. Concurrent with those studies, Bernardo Franklin, analyzing some of the results of microarray data that we from patients whom we had studied in Porto Velho, Rondônia, discovered that many of these patients had genes that seemed to be induced by a DNA-like molecule. That is to say, we know from viral infections with DNA viruses that patients produce lots of type 1 interferons, and when we looked at the cells from these patients who came in with malaria, they also seemed to express genes that were downstream from type 1 interferons. Genes such as AIM2 and IFIT-M4P are genes that are thought to be relatively specific for type 1 interferon activation. That led Rosane de Oliveira in the laboratory to look at mice that failed to respond to type 1 interferons. These are mice with defects in alpha-beta receptors. And here you can see that these interferon alpha-beta receptor knockout mice have no problem when they're infected with a type of plasmodium that causes cerebral malaria in mice, but wild-type mice succumb rather rapidly. In this experiment, she's looked at about 40 animals. This has now been repeated 5 times in 2 different types of genetic backgrounds, and we're very convinced that this is true. So we began to study type 1 interferons in hemozoin and DNA. This was the first experiment done by Peggy, where she took peripheral blood mononuclear cells and incubated them with hemozoin, or hemozoin that had been digested with nuclease, or LPS as a control. You can see the hemozoin very nicely activates interferon beta mRNA production, and that's DNA dependent because it goes away after DNA treatment. I'm not sure why that slide is so out of focus, but if instead of using DNA, we do the same experiment using our oligonucleotides that contain this AT-rich motif, get the same type of results. Here we're using AT2, stimulates PBMCs to produce interferon beta, and this is Sendai virus that's used as a positive control. Neither the AT-rich nor the plasmodial DNA that we use to do these experiments required a toll-like receptor. And this was a huge surprise to us. It's totally toll receptor independent. Well, why do we say that? Remember the adapter molecules I showed you earlier? We have mice that are missing MYD88 and TRIP. Those are the 2 adapter molecules. If you knock them out, you lose all toll signaling. So when we did the experiment comparing wild-type mice compared to MYD88 TRIP double knockouts, we found that both nucleotides containing an AT-rich motif and DNA from the genome of Plasmodium falciparum could induce a type 1 interferon response. On the right side here, we're looking at a control, CpG DNA, which activates Toll9, and you can see that that response is ablated in the knockout mouse. Now there are a variety of known receptors for DNA, dye, RNA helicases, and RNA Toll3. I'm not going to go through these individually, although I think I have the data, but I'm just going to say that we looked very carefully to see whether this activation was through the known DNA receptors, and the answer was, it was not. So we'll pass on these. Now, type 1 interferons are activated by transcription factors, and the most important of these are the IRFs. And we have knockout mice that don't have IRF3 or IRF7. So Rosane Oliveira went back to the laboratory and she tested mice that had double lesions in IRF3 and IRF7 in comparison to C57 black 6 mice. This, again, there's been a little bit of a PC to a Mac problem here. But these are 2 experiments with these knockout mice, and you can see that compared to the IFNAR knockouts, the IRF3, IRF7 give the same type of results. So this signaling that we see, incidentally, in using oligonucleotides or genomic DNA from Plasmodium falciparum is exactly mimicked by what we see in terms of the outcome of cerebral malaria in the mouse. Now, the AT-rich DNA, in order to stimulate cells, has its ability to do so only in the cytoplasm of cells, not in the endosomal compartment like the CpG DNA. And the question is, how does it get there? Now, experimentally, it gets there because we mix the DNA with the transfection reagent. But of course, in nature, people don't get malaria and then an infusion of a transfection reagent simultaneously. So we think that the way it does so has a lot to do with hemozoin. The hemozoin is responsible for moving the DNA from the phagolysosomal compartment into the cytosol. And how does that occur? Well, we know from work done studying another type of innate immune response, the inflammasome response, that phagocytosis of inert particles results in phagolysosomal leakage. And this has been shown formally and reported for crystals such as silicic acid, which causes silicosis, urate, which causes gout, asbestos, which causes asbestosis, the protein beta amyloid or the peptide beta amyloid that's associated with Alzheimer's disease and dementia, and as I'll show you, hemozoin as well. All of these, incidentally, are activators of what's known as the NLRP3 inflammasome. So here's an experiment done by Parisa, where she's taken hemozoin and incubated it with cells for a couple of hours. You can see by reflection confocal microscopy that it nicely gets into cells. Then she took the cells and loaded them up with fluorescent dextran. Now, fluorescent dextran is a molecule that typically goes into the lysosomal compartment, and here we can see in cells that it's very nicely contained in lysosomes. But in cells that have been pre-treated with hemozoin, these lysosomes have lost their membrane integrity. So all the dextran gets into the lysosomal compartment and freely flows out into the cytosol of cells. And we think that's what happens with the DNA on the surface of hemozoin. That the DNA carries it into the lysosomal compartment, like I showed you earlier, that the phagolysosomal compartment is filled with enzymes that chop up the DNA and release it from the surface of hemozoin, and that gets into the cytosol where it can activate a cytosolic receptor. So that's shown on, that is reiterated on this slide. As a result of phagolysosomal membrane deterioration, malarial DNA can move into the cytosol and activate one or more nucleotide receptors, resulting in a TLR9 independent cytokine production. And this is shown in this slide, where Parisa has taken cells and incubated them with hemozoin. They're coated with CpG oligonucleotides. And here in the overlay, actually, you can see that in time, trying to get to this, if we blow this up here, we can see that in time that the hemozoin moves out of the lysosomal compartment, which is blue, and it's the contents of the phagolysosomal compartment and do similarly to activate the cytosolic receptor. So this is just the last slide. This is a recap of everything I've told you. First, the pro-inflammatory response to B. falciparum infection is not solely due to GPI anchors activating toll-like receptor 2. Second, there appear to be two major ways that the human host responds to plasmodial DNA. The first is as DNA traffics through the lysosomal compartment, it rapidly engages toll-9 via CpG motifs. The second is that when DNA gains access to the cytosol, an AT-rich motif activates an as yet unknown receptor via these intermediate molecules that I didn't talk about, known as TPK1, IRF37, and STING. And finally, hemozoin promotes innate immune activation. It does so directly by activating the inflammasome, which I didn't show you, and indirectly by carrying DNA into the lysosomal compartment and allowing the DNA to escape afterwards to the cytosol. So these are the people who have done the work. First, I want to mention that in Worcester, that we have a very close collaboration of a group, or we call it our malaria group. It involves Kate Fitzgerald, who's a world-leading expert in interferon regulation, and Ricardo, and myself. These are some of the students. I've mentioned them all in passing when I showed you their experiments, in particular Shruti and Parisa Bozani, whose experiments I showed you in some depth. We've had many previous students who've helped a lot. Peggy Peroche, Bernardo Franklin. We have lots of other collaborators with whom this work couldn't be done. In particular, one of them sitting in the audience here, Dr. Luis Herrera, see if I can get this right, Luis Librando Pereira da Silva, who has graciously let us work in Porto Velho with his patients there. And we also have collaborators from all over the world who give us knockout mice, including Shizuo Akira, Tadagatsu Taniguchi, and Glenn Barber. And finally, we now have an emerging collaboration with a drug company, Eisai, in the Boston area. And I think Ricardo's going to tell you about work that we're doing with them to try to come up with some immunologic cures for cerebral malaria. Thank you very, very much for your attention. I'll leave that off just in case some of you want to come to Toll. Incidentally, we would give a discount for a lot of Brazilians to this meeting. We always have a lot of Brazilians at the meeting. And you don't have to be in biology to come.